Thank you so much for agreeing to be a part of the inductive economy, Dr. Vatsala. It's an absolute thrill and a privilege to have you on the show.
Thank you, Vignesh. It's my pleasure to be speaking to you and to your audience.
Thank you, ma'am. Before we get into the bulk of your work, and we're going to be covering your perspectives on the future of science and research in India. I really want to touch upon something that might be a little arcane to our listeners, but I think might be very close to your heart. So, can you talk to me a little bit about the work and the effect of this gentleman's work has had on you, Mr. Bagyam Ramasami? And can you talk to us a little bit about him?
Absolutely, absolutely. I mean, it's wonderful that we can start the conversation with Mr. Bakim Ramaswamy because he is an author. He's a Tamil author. For those of you who don't know him, his original name is Jalakantapuram. Oh, sorry, I'm blanking out on his first name. But I know him as Bakyam Ramasamy because that's the pen name in which he wrote, and he wrote so many funny, humorous stories in Tamil. But mainly, like PG Wodehouse, he had this main character, couple of main characters, Apusami and Sidapati, an elderly couple, and they are so funny. I mean, the characters are funny, the stories are funny, and every time, so Sida Pati comes out as the better one, and Apusami always loses out to his wife. The various schemes that he does and all that. It really, my summer holidays in school and college, I would go back to these books and read them again and again and again and keep laughing. Every time I read it, I laugh. So he's brought incredibly a lot of happiness to me and continues to do so even though he's not alive anymore.
Absolutely. Thank you so much for that answer, ma'am. And, diving in, moving forward, just want to ensure that, our listeners have the philosophical and conceptual foundations of your work in place. So, my first question is, in the grand tapestry of life, movement stands out as one of the more fundamental expressions of biological existence. From your perspective, what is the deeper significance of studying neural circuits that control movement?
So, if you look at life around you, right? I mean, there are plants and then there are animals. One of the things that happened is that when organisms became from single celled to multi celled, where they could make much more complex arrangements of cells, um, somewhere along the way, it became critical to be able to move around to find food, partners, escape predators, and so on. So it added evolutionary fitness, so to speak. You're no longer bound by the conditions of where you happen to be, right? So that increases your likelihood of survival because now you're not limited by the resources that you can find in your local environment. And therefore movement is an evolutionary advantage that happened, that enabled a large class of animals to be able to find newer niches in which they can survive, right? And but to be able to move is not so easy. You need a lot of different things working together. First of all, the physics of movement itself, whether you're in air or in water or in land, how you move is defined by the physical properties of the substrate where you happen to be. So there is the physics aspect of that. But to be able to deal with that substrate property, you need to have mechanical properties of the animal itself, such as muscles that can produce force, which will allow you to navigate that space, right? So you need to have the machinery. So okay, now we have the machinery, but that alone is not sufficient. How do you control this machinery? And that's where my work comes in, which is that to be able to understand how is this complex machinery of the body controlled by the nervous system. And the more complex organisms got, the more complex the nervous system organization to be able to control movements. And therefore it's purely a curiosity based research that I started, but it has a lot of different applications as well, in terms of diseases that affect our ability to move, applications in robotics, for example, although I have not done anything in that aspect. But it does have those sorts of vignettes as well, that if you understand more about how animals move, you can perhaps use those principles to be able to build better robots that move around and navigate the space around them. So those are sort of the big picture. But the reason I got into it was not first at first sight to be able to understand movement. My question was even more basic. I wanted to understand how any piece of nervous tissue functions, and it's perhaps the best studied in the context of locomotion, because in the context of locomotion you at least know what it is that the output is supposed to be. Something's got to move, right? Whereas if you're studying, for example, memory or learning, the outcomes are a little bit more abstract, and the processes that need to go into those kind of process, into those kinds of outcomes, is also more abstract, and it's difficult to have an animal report to you whether it remembers something. Although there are assays, but it's much more complicated. But with movement, any model organism that you see, you can assay how much it moves, how it moves, what kind of limbs it moves, and so on. So the output is much more tangible compared to other more abstract kinds of output that the nervous system produces. So this was my immediate goal, to be able to have an output, and which is movement, and then to be able to understand what it is that drives that output. So defined output, what is the input that goes into it, and what is the computation that goes into it. That's how I got into it. But I've become more interested into the finer aspects of locomotion itself. But I still see motor systems as sort of a proxy for our broader understanding of what the brain does in a larger sense.
Right. And your work on CPGs really offers a glimpse into that, elaborate neural orchestration, right? So just to ensure that, we call this phase the philosophical and conceptual foundations of your work, right? That's the segment. So just for the benefit of our audience, can you talk to us a little bit about, what are central pattern generators or CPGs to kick the
Sure.
conversation off?
Yeah, so CPGs, or central pattern generators, are neural networks that are capable of producing rhythmic output. So, why these are special is that this is a piece of nervous tissue that is able to produce its own beat. Even if you take away all of the other inputs to it, it can still sustain its own activity, and it produces oscillations. Yeah. So this was sort of an enigma. How does, because earlier it was believed that the nervous system or the brain is simply reactive, that sensory inputs, when we see something, when we hear something, something gets into the brain, and then the brain is triggered into action, right? So action was always thought to be, this was about more than 120 years ago, that people thought was that all action was actually a reaction. It was a reflex, that some sensory input comes in, triggers a piece of the brain to react and produce walking, or running, or whatever. But experiments in the last century have clarified that you can get rhythmic activity in the nervous system, even when there are no timed inputs coming into the nervous system, right? And this is best seen in invertebrate preparations. So you take, for example, a lobster or a crab. This is what I did as a PhD student. And you can actually cut out the nervous system from the animal itself and put it in a dish containing seawater, and the nervous system will continue to produce oscillations. It will continue to produce activity that is beautifully timed. And so this tells you that there are mechanisms within the brain that are self-sustaining, right? And why this is important is that if you look around you, all locomotion is kind of rhythmic. You take the slithering of a snake, or the flying, the wing beating of a bird, the walking of a human, the galloping of a horse. All of these will have some sort of rhythmic pattern that repeats again and again and again. Like in the humans, it's left, right, left, right, left, right, right. And this is made possible by the CPGs in our spinal cord. We have these circuits in the spinal cord that can produce these left, right alternating commands to the muscles. So when you're walking, these CPGs tell, okay, left leg, your this muscle is going to contract now, your that muscle is going to contract now. So it keeps on repeating, and you're able to do that. And if you understand how these CPGs work, which again, we've not gotten anywhere close to a complete understanding of how these CPGs work. And the other major CPG is in the brainstem, controlling your breathing. That when you breathe in and out, in and out, in and out throughout your life, these CPGs are going bum, 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 bum. I mean, just like the beating of the heart, right? So this is very interesting, because how do you make a piece of nervous system produce these kinds of rhythms? And the rhythms are not like, set in stone. They are very plastic. You can make them change in frequency, in phase relationships, and so on. Like for example, you see dogs on the street when they are trotting, when they're walking slowly, they'll have their legs alternating. And then, if they're chasing someone, they're going in full gallop, right? And then the relationship between the legs changes. So this is called gait change in neuroscience, and the gait change is something that the CPG does. So it's incredibly reliable and repetitive and rhythmic and all that, but at the same time, it's not set in stone. So how do you do that, right? This is again, it's a question for mathematicians, it's a question for neurobiologists, and it's a question for clinicians, because you can use these mechanisms if a person suffers, let's say, spinal cord injury. So you might ask me, okay, you're saying you have these CPGs in the spinal cord, why do people become paralyzed when they suffer a spinal cord injury, right? And that's because even though we have these CPGs that can go repetitive, they still need to be instructed by higher brain centers when to start, when to stop, balance. So we are upright animals. We need balance to be for the CPGs to be able to drive these leg motions. So balance needs to be kept. All of those things are still supplied by the brain. So when you have an injury, those mechanisms are taken away, and that's what results in paralysis, because even though the machinery is there, nobody is there to tell it how, what to do, and when to start and when to stop. So it just goes silent.
There's a really profound significance from an evolutionary standpoint, the way CPGs have really influenced our own existence as species. And can you tell us, probably, from your, given that you're given your unique vantage point, the evolutionary origins of CPGs? How is it that they were selected to give us mobility as a species?
Yeah, so CPGs, you can scientists of course have studied them in a wide variety of organisms, but there are certain fundamental features that have been conserved. You can take CPGs in a leech, for example, which is an invertebrate animal. Doesn't have a vertebral column. And you can take the CPG in a mouse, which is a mammal like us. And there are some fundamental principles which are conserved. In fact, much of what we understand today about CPGs comes from studies in invertebrates. So it's a very very early evolutionary invention. Having we've seen it from in lower invertebrates all the way up to primates. So it is definitely useful for controlling rhythmic activities. Now, what are the conserved features that I'm talking about? Um, for example, if you have um, a set of neurons, right? And you want to make a circuit with which you want to make rhythms, there are many ways in which you can do that, right? You can make each one of them capable of oscillations, right? And then you can hook them up in a way that all of them oscillate together. right? And this would be like a group of oscillators that synchronize themselves. Or you can make it such that individual neurons are no longer oscillatory, but you can make them like a cycle pedal. If you press on the cycle pedal, one pedal goes up, and the other pedal, if you push on the pedal and make it go down, the other pedal automatically comes up, right? And then you push on this pedal, the other pedal comes up automatically. So like that, you can hook up two neurons such that when one set of neurons is active, the other set is inhibited or it goes down. And then when this set goes down, this other set comes up, right? So these are called half center oscillators. So you can build networks like that. You can build networks wherein, there's no rhythmicity at baseline, but you endow it with some trigger, and then the trigger makes them oscillatory, right? So these are also present in our bodies, in our nervous systems. So these are called conditional oscillators. So all of these different combinations are present, and you can see them in organisms wide ranging from invertebrates all the way to vertebrates. And it's obvious that we keep across evolution seeing these mechanisms pop up again and again in various places. And recently there's some thought that says that not only for locomotion, not only in spinal cord for driving movements, maybe CPG mechanisms are present elsewhere in the brain also, such as in the sequencing of thoughts. When you're thinking about episodic, when you're remembering things, when you're remembering episodes or places, one is hooked to the other, is hooked to the other. So it's like a chain of events. And that is perhaps like a CPG, which, brings up these different events with a defined periodicity. So there are these abstract ideas about how CPGs could be used in other places as well, but that's roughly the outline of what we know thus far.
So it's really interesting that we are around 10 minutes into our conversation, almost 20 minutes, and you've made multiple references to the word rhythm. So there is a very colloquial understanding of what rhythm is, and there is a very specific definition of rhythm when you use it. So can you unpack that a little and probably explain the differences or the points of convergence?
Absolutely,
Yeah.
absolutely. I mean, we understand rhythm in the context of music. So even in music, we have the bass, the beats, right? And then you have the notes of the song itself. Like that, living organisms also have rhythms, and it's not just the rhythms that I'm talking about. There are many kinds of rhythms that are there in living organisms. One thing that your audience might be very familiar with is circadian rhythm, which is our daylight cycle, where we wake up at a particular time, we eat at particular times, and then we become tired and fall asleep in a certain window, and so on. So apart, and circadian rhythms are also governed by rhythms in the brain. But apart from that, the brain has a lot of other frequencies, in which it beats, in which it oscillates, and it's like a symphony that the neurons are playing. And neurons talk in language of electricity, and they have these tiny blips of voltage signals. And these signals are typically in the millivolt range, one in a thousand of a volt. So one in 1000 of, or less than one in 1000 of a AA battery, for example. So they are small signals, but when a whole group of neurons signal together, then it becomes a symphony. It becomes a large enough signal that multiple regions in the brain pop in and talk in these rhythms. The rhythm that I am talking about is a rhythm that is generated in groups of neurons again. And like I said, these rhythms are directly responsible for the rhythm of locomotion. When we are running or when we are walking with a certain speed, that speed is determined by the rhythm that the spinal cord generates. So it's an oscillation in the membrane potential, in the voltage of the neurons, which again is dictated by properties of the neurons themselves, which are each neuron is special. I mean, we have about 100 billion neurons in our brain, but one neuron is not like the other. They are very, very unique in multiple aspects. Multiple people have tried and failed to group neurons, and they find that one more criterion, and they bring in, and then the neurons fall apart. They don't stay in the same group. So every neuron is unique, and that uniqueness is what makes them generate these unique activity patterns, these rhythms. But they can synchronize when they want. They can uh, pop out of the band when they want. These are all things that neurons do all the time.
Right. And thank you so much for that. And I'm very curious to sort of have a, I wish I could be I wish I could be a fly on the wall the moment you realize the profundity of the profundity of the implications of actually having CPGs in not just movement, but the rhythm activities, and the ability to even be locomotive species. So, can you talk to me about when you had that breakthrough, and or the profundity of it, the moment you sort of experienced it, all the way back in time?
So the moment when I realized that studying CPGs is a window into the
Yeah,
brain, I actually don't remember exactly that moment, but as I said, when I went to grad school, I had very little idea or notions about neuroscience, um, and I was pretty much set to go to a lab which was studying cortical neurons in culture. So what they would do was take these neurons from mice and put them in tissue culture, and look at their how they react to long-term changes as a model for how our brain would respond to long-term changes, right? For example, if a person is in depression over a very long time, how does that affect their brain? Or if a person is a chronic epileptic patient, how does that affect their brain? So this is what they were studying. So I was pretty much set to go to that lab, and I went to a computational neuroscience lab to do a short rotation. And this lab was attached to the lab where I did my PhD thesis. This is Professor Eve Marder's lab in Brandeis University. And I had no idea what this lab was doing, except that they were using lobsters. And at that time, I had not seen lobsters in real life before I got there. These are the clawed versions, but I sort of thought that these are like slimy sea creatures, like sea slugs. I had no idea what they were and what they would look like, and why even people would use them. One afternoon, I just walked into this lab, and they were doing studies on baby lobsters. They were looking at the development of networks that control movements using these baby lobsters. And if you look at these baby lobsters, they are really like that tiny, and they are sort of brilliant blue in color. And you see a little miniature creature which is blue in color. It really makes you fascinated. So I stayed there and watched what they were doing, got talking to them, and it turned out that they were looking at the movements of the stomach of the lobster. So these lobsters have teeth within the stomach. And being scavengers of the ocean, what they do is they just quickly grab, like crows we see outside. They maybe we should call them crows of the ocean. They quickly grab whatever is available, dead, rotten material, dead animals, whatever they can find, and stuff it in their mouths. They don't take the time to chew them. And once it goes in the stomach, then the teeth in the stomach, inside the stomach, the teeth start cutting and grinding the food. And there are muscles that control how this stomach crushes and grinds the food. And there is a tiny nervous system of only 30 neurons that controls the stomach. It's a separate nervous system. It's separate from the brain of the lobster. Um, so I got to understand more about what they were doing, and that is probably when I thought that this is a real neural network, that is functional. You know what the function is, you know what the and the beauty is that it's only 30 neurons, right? So it's like, suppose in the future, we have more advanced humans and they come across our computers and they don't know what these things are, and they also come across a calculator. Maybe it's more simpler for them to understand how a calculator works before they understand what a computer works. So it's something similar. The nervous system in the lobster that controls the stomach with only 30 neurons is so much simpler compared to the human brain, and therefore it offers a real possibility that you can break apart this thing to its limit and understand how each part works and what happens when you put everything together. So that was the hope with which I started in this field, and that's when I got interested in CPGs. My main interest was to understand networks of neurons, as I said before, how when you put groups of neurons together, how do they do something so intelligent? How does it build up to more than a sum of its parts, right? So that's how I got into this business of, from the lobster stomach, is is how I got into this field.
And it's really interesting that you use the words how I got into this field, because your background is actually as an engineer, and I think you were one of the earliest batches of biotech, I think out of, yeah, the first batch of biotech grads out of, I think, Anna University, if I'm not mistaken. And it's really interesting that you went from something that's as, I always really think of this physics example, that there are a lot of great theoreticians who studied pure physics, that they went on to study pure physics in their PhD from their undergrad. And then there's somebody like a Paul Dirac who had the engineer's mathematical skills. So when he would do physics, it would be with the precision of an engineer, and there would be no major meanderings or anything. It was very clear in what he wanted to achieve. So when you go from something as disciplined and applied as engineering and biotechnology to something a little more, if I may be allowed to use the word, abstract or discovery oriented, where you have to figure out the first principles for yourself and they are not written down in stone, for example, in neuroscience. Can you talk a little bit about that transition and how you had to sort of unlearn certain mental models and learn some new mental models or relearn mental models? Can you talk about that?
Absolutely, absolutely. I mean, that's a very good point. So one of the things that, as a biotechnologist, you learn is, the what do you, the crystallized nature of concepts, right? For example, you learn about how DNA is replicated, or how, you can cut and paste pieces of DNA. It's almost like playing with Lego. It has a certain definitiveness to it, right? If a restriction enzyme is supposed to cut DNA at a particular place, it will cut at that particular place. And you can see it on an electrophoresis gel. You take a big chunk of DNA, and your restriction enzyme is supposed to cut it into two pieces, this long and this long. Then you put it on the gel and run it, you will get a band at that particular spot where you expect it. So there is a definitiveness in, in terms of dealing with molecules and dealing with, how biology works at that scale. Molecular biology, and genetic engineering or genetics even. When you have these, when you study about monohybrid crosses and dihybrid crosses and the ratios with which Mendel got his different kinds of peas and all that, there is always a determinism to that. The one thing that really boggles my mind about neuroscience is that there are a few places of determinism, right? For every example, there's always a counter example. Not to say that there are, fundamental principles by which things act. There are very many biological concepts which are deterministic, but then, you go up a little bit in scale, then you get all of these complexities that just, blast out of the box. Let me give you an example, right? This is something that I teach in my class as well. You take a cell, okay, a neuron, which makes action potentials at a regular rate, right? Um, I mean, this neuron could be making these action potentials at this regular rate when it is um, under a certain group of conditions. But the minute something about its environment changes, it can change its activity, right? So you can't say that, okay, this is a neuron that only does this unless you've sampled it for its whole lifetime. Um, or you take a certain group of neurons that are, let's say that have inputs coming into them, certain groups of synaptic inputs coming into them. Those inputs can change in their strength. You can get very strong inputs under certain group of conditions. You stimulate something, you can make those inputs go smaller or bigger, and this is synaptic plasticity, right? So this plasticity in the nervous system really makes things more complicated. Plasticity means that ability of things to change, just like a plastic can be molded into things. The nervous system is plastic, and so what is today is not what is there tomorrow, or what is today in a under certain groups of conditions is not the same as what it is tomorrow, right? And so this is something that's unique for the nervous system, and something that's also makes it much more complicated than other fields of biology that I have encountered. uh, does that answer your question?
No, absolutely. And I was just making notes on the side, and one of the questions that just came up was, given that you've gone from applied to theoretical to, really deep research, and it takes a lot of understanding and learning and relearning. I was just very curious to know, what's the most elegant theory that you've ever come across, in so many years of study and research. I've, yeah.
Yeah. Theory with evidence or without evidence?
I think you should answer both. I think that's what makes it fun. Yeah.
um, so the theory with evidence, let me talk about that first, is easier. The theory with evidence, which is something that has influenced me deeply, is the fact that the nervous system produces function. It can produce the same function with very different properties, and vice versa. It can produce different functions with similar properties. This is called degeneracy, or this is also called redundancy in the nervous system, where it's got both resilience and plasticity, right? Stability and plastic, two sides of the coin. You need, see, the nerve cells in our brain, the neurons, they are post mitotic, meaning that once the nerve cell is differentiated from stem cells, when we are born, initially, when the embryo is growing, there are stem cells which make the neurons. But once it becomes a neuron, once it takes on the identity of a neuron, after that it doesn't divide. Okay, if you want new neurons, you have to go back to the stem cells, which is why we hear all this talk about stem cell therapy for neurodegenerative diseases and all that. You can't make more neurons from one neuron, and right. So you have to have, when you make that neuron, you have to make it such that it lasts, in the case of humans, for about 70-80 years, right? And it continues to do what it does with stability. But at the same time, we also see that a human goes through a lot of changes through their lifetimes, right? The initial 15 years of development, of acquisition of language, for example, cognitive capabilities, locomotion, in the early years, trying to crawl and then stand up and run and all that. And then later on in life, where the body is also changing, right? And the brain still has to be able to, from controlling a small 1.5, 2 feet long body to a full blown human to be able to control that body. And then the changes that happen, such as in the case of women, for example, puberty, pregnancy, menopause, where the hormones are circulating wildly, very different levels of hormones, and yet the brain has to be functioning as it is. So you need that stability, but at the same time, it is learning new things. Every day we acquire new memories. Every day we are changing. I'm not the same person I was 5 years ago, right? I'm changing as a person. My emotional response is changing. So you have limited hardware, right? These neurons that you're born with, that you have to use for the rest of your life. And that limited hardware is used for both stability as well as for plasticity. And we are only beginning to understand some parts of it. As I said, I started out with a grandiose statement that this is a theory we understand. We are beginning to have clues about it. Let me be more modest about it. We are beginning to have clues about how this may happen. um, but this is, something that struck me as being quite powerful in terms of having a system that does both. um, so that's one theory that I can talk about. um, the other theory has to do with development in terms of how the nervous system is put together. So again, here these are both theories that have little bit of evidence, but you know we need more work to be able to put them on solid ground. So there is a raging debate in neuroscience called nature versus nurture. And it is my humble submission that it's probably both, but nurture probably has more to play than nature, because of the extended time period over which we have nurture compared to nature. What is nature and nurture? Let me first explain that. Nature is our genetic makeup, in terms of what our genes tell us who we should be. And nurture is, of course, everything else that comes in from the outside, the kind of opportunities we have, the experiences that we face, the nutrition that we get. And of course, when the baby is in the womb, what kind of experiences the mother goes through. That is also nurture, because the any sort of stress or trauma that the mother goes through gets passed on to the baby and affects their nervous system. So nature and nurture, again, two sides of the coin. And both have a role to play in terms of how the brain develops. But I would stress that we need to understand more about nurture than we do about nature. There is probably, relatively speaking, less variability in nature compared to nurture.
Understood. And super interesting that a lot of these insights basically come from your exposure and a lot of the research that you've done. And one of the principal organisms that you actually model is the zebrafish, right? So, can you talk a little bit about, what are the advantages of working with zebrafish, or what does it actually have to teach us about the universality of neural mechanisms across organisms?
Absolutely. So, zebrafish is a small vertebrate animal. It's actually, its home is in India. When researchers first identified it as an organism with which they can work, they actually took the sample from Darjeeling and took it back to Oregon, where they, grew it and, worked out conditions for it to be used as a lab animal. So it's actually very much Indian. The bad news is, of course, that this was the thing that I talked about when they took it from Darjeeling. That was done in the 70s. And the habitats that they found it in the 70s, those habitats don't exist anymore. You can't find zebrafish there anymore because of pollution in the water bodies and whatnot. But having said that, why do we use zebrafish? So first of all, it's a vertebrate, unlike the lobster that I was talking about. So in the animal kingdom, if you take vertebrates and invertebrates, there are a lot of differences in terms of how their body is structured. We call it body plan. And similarly, how their nervous system is structured. If you take all vertebrates, the nervous system is largely divided into forebrain, midbrain, hindbrain, and spinal cord. And so this organization is present in the zebrafish as well. So you have a forebrain, midbrain, hindbrain, and spinal cord. You have similar types of neurons between, if you compare mammals to zebrafish. You take any piece of the brain and you look at the circuitry, right? So the brain works pretty much like a computer because there are specific connections between neurons. It's not that they connect randomly. Sometimes a neuron will send an axon over several hundred millimeters to reach its target, right? It won't make any connections locally. It will try to talk to somebody quite far away. So that means it knows where it wants to go and who it wants to talk to, right? And this same circuit layout is present in the zebrafish, right? So these things are conserved. So when people initially started working with zebrafish, it was not as a neuroscience model organism. They used it for understanding early development, and the attraction for this was that the embryo was transparent. It's like a glass marble, so you can put it under a microscope and watch as the cells divide and as they make an animal, right? And so because of the homology with mammals, where I said that the body is similarly structured, the genes are conserved. There are, the genes that you find in humans are similar to the genes that you will find in zebrafish. There are always, homologs between the two. So that means that if you want to understand early development, which is not possible in mammals, because in mammals the early development happens in utero, inside the uterus, where you can't observe it. I'm talking about development which is like from the single celled stage, when the egg and the sperm have fused, they formed the single cell. After that, what happens? What are the processes that happen? How is it that an animal is formed? Those kinds of processes are accessible in the zebrafish because it's transparent, and the fertilization happens outside. So the male fish and the female fish come close together, and then the female drops the egg into the water, the male releases sperm into the water, the fertilization happens outside. You can collect the eggs, put them in a dish, put them under a microscope, and watch as these cells form. They divide and they divide and they make these germ layers, and then they make the whole animal. So that's why they've brought the zebrafish to study these early processes of development. But somewhere along the 1990s, people realized that, this is a good model organism for studying other organ systems as well, because not only the egg but also the larval stages are all transparent. You can see the brain. And then in the 2000 something magical happened, which is that people made genetically encoded calcium sensors, which are proteins that you can engineer into the zebrafish and watch as the neurons do their work. So you're not only just watching the cells, you're also watching what they're doing. When they become active, you can make them glow brighter when they become active. So you can watch this neurons in action while the fish is doing something. So these breakthroughs, one after the other, over the last 50 years or so, is what makes the zebrafish as a powerful model organism. But also what makes it powerful is that we can insert electrodes into the brain, and we can sample the activity of single neurons. So there is no skull in the early stages. So you don't need to do surgery that's painful for the animal. You let the animal be. You're not, doing invasive procedures. It's just a skin on top of the head. You can paralyze the animal using drugs, and then you can insert these fine needle electrodes and assay the activity of single neurons, and you can see what the animal is doing. So you go all the way from single cell to a whole animal behavior. So that makes it really powerful to study these organisms as a model for neuroscience.
Absolutely. Absolutely. And I'm really curious, since now we are on this track of actually conducting the research, I want to sort of move it to the next segment, which is, what might be the ethical considerations when we
Mm,
start studying more complex organisms to understand what are the principles that underlie their locomotion in this very specific case. I think it's very interesting that the applied sort of um, understanding of the principles that come out from, say, for example, when you run experiments on C. elegans or when you run it on zebrafish, right? You sort of extrapolate it and you do a little bit of engineering work. It's the way that our scientists sort of do rocketry today, where they
yeah.
take these American sort of 50-year projects and then they
Mm.
just engineer it down to the nuts and bolts and they release it at a third of the cost and suddenly it's like performing, right? So
Yeah.
what are certain ethical considerations that you have in mind when it comes to charting these unexplored organisms and studying them?
Right. So, yeah, I mean, all biologists have to, whoever uses model organisms, do have to clear their experiments through an animal ethics panel. And we try to minimize the pain to the animal, and we try to reduce the number of animals used, and we try to reuse the animal whenever possible, so that we are not using animals again and again. But also, every experiment is evaluated in the sense of whether this experiment is necessary to do, right? In terms of the ethical questions, I think there are two parts to it. One is, the ethics of using the animal itself, and two is, what are the boundaries of neuroscience knowledge. What kinds of questions would lead to an ethical dilemma, right? For example, if we had the capability to read the thoughts of a human, would that be ethical? Should we even research that? um, for example, there's another thing, which is that we've many labs around the world have started making organoids, which are a small organ in a dish, which is why it's called an organoid, where you can take a ball of cells, differentiate them into neurons, and they'll form a small nervous system in a ball. And multiple studies have shown that these organoids can be made in such a way that they replicate a part of the human brain. They have those layers, they have the specific connections, the specific cell types, and so on. Of course, it's just a ball of cells, but when does it stop being just a ball of cells? Right? Do we know the answer to that question? I don't have an answer to that question, right? um, but the where do we weigh in the trade-offs, the pluses and the minuses of doing such work? On the one hand, yeah, there are biomedical benefits, which is that people suffering from different kinds of diseases, neurodegenerative diseases, um, they will benefit from it if we are able to make a piece of tissue that can integrate with their nervous systems and help them regain function. But on the other hand, if it's a ball of cells, how do you tell whether this ball of cells is not awake, right? And what it's thinking, right? Is it even thinking? These are very provocative questions. It's the limit of, it's like, AI, for example. Today we have debates about AI, ethical AI, and so on and so forth. What is ethical AI? But then AI can't be more ethical more ethical than the training data set. If the training data set is itself biased, which is us humans, it's on us, how can you expect AI to be more ethical than that, right? Or how do you even teach something to be ethical? So these are at the edge of our understanding right now, and I hope we get more clarity. But coming back to questions on animal usage, yes, I firmly believe that we should only do experiments on animals where the questions it really do require an animal model. And even for those questions, it should be less. We should limit the use of primates and mammals, for example, Not that the other organisms don't feel pain. All animals feel pain, they have pain pathways. But I think, in terms of our ability to empathize and understand their feelings, we probably understand pain and suffering in organisms which are closely resembling to us, compared to, let's say, a larval zebrafish or a drosophila fly.
Yeah.
Um, so and so we are much more comfortable using them for experiments. um,
I am incredibly curious here because you brought up a lot of things that I want to touch upon. So here's one of my pet hypotheses. Like, I really like to ask people this. Like, Roger Bannister had the 4 minute mile, right? Like, people didn't know if you could break the 4 minute mile, and then Bannister did it, and then people began breaking it after that, right? So when it comes to AI, the kind of way in which I look at the Turing test is like that. So the Turing test for a long time had been the 1 minute mile or 4 minute mile or whatever, right? So for many years, many decades, you did not have a breakthrough. Then you had ImageNet, and you now have Chat GPT, right? Now all of a sudden you have Claude, you have so many of these other models, these foundational models, which are coming and just knocking the test out of the park, right? So much so that there's, I think, been a study in some paper which I read, that there is actually a subset of people, consumers, who prefer AI generated content over human generated content, right? Which I thought was really interesting. So the question is, great, a Turing test or a 4 minute mile was a benchmark at a certain point. Once it is broken, what is the next equivalent of it that actually comes? I think that sort of then becomes the next benchmark for a lot of people to break. So that's I just wanted to share that perspective since you raised the AI point. The second thing that I wanted to sort of ask was, in terms of the reliability or the usage of animals in running these tests, one of the problems you will have is who becomes suddenly the arbiter of what's valuable and what's not valuable, right? So you were talking about tissue plated computing or something like that, where if you could just get into the tissue and you have it, then you can have models and you can simulate it and you can run it. So the question I wanted to ask is, if what would it take to get you the simulations of animals that you actually want to test things on, so that you don't have to rely on the real live animal to actually see what
hmm,
the outcomes are.
hmm. So, yeah, this is a great question. And one of the things that's happening in the field is that there is increased sharing of data. And data sets are now being open to, the entire world. So I don't need to replicate an experiment that was done elsewhere. For example, the electron microscopy data sets. So you take a brain and you slice it up into very thin slices, and you capture a picture at the electron microscopy level, so you can really see the very fine structure of the nervous system. And this is an expensive experiment. It requires a lot of hardware, and it then, people can reconstruct pieces of this brain. Where does this neuron project to, who it connects to, and so on. But now this is being crowdsourced, so there are people who can, just for the fun of it, get into this system and trace out connections, right? So it sort of democratizes science, that it's not the places where, which have a lot of money for a big electron microscope. Earlier it used to be that whoever has the money for the hardware, the equipment, which costs a lot of money, those places had the edge over all other places which were resource deprived. And this kind of science that came from these two kinds of environments would also be very different. Third world countries typically would be ones which could do only resource limited science, whereas the first world economies could do invest in long term, lot of equipment heavy science, consortia, and so on. But with now the sharing of data, things are becoming more equitable. I wouldn't say it's a level playing field at all, but it's getting there. I hope it happens more and more. Because now people are also required to share their data sets. You can't hold, you publish one paper, you're required to share your data set so that other people can mine this data set and do the kinds of things that you talk about. Like I can take data that's in a public repository and make models and answer my own questions. So that's definitely possible these days. And there is a repository called Neuromorpho, to which we've also contributed, which contains a library of neuronal structures. And so people can go there and pull out their favorite kind of neuron and see what are the different structures that people have put in. So these kinds of things are happening.
So I think I know which website I am visiting after our conversation. So I think that's great. So thank you so much for sharing that. I think one very interesting way that I'm sort of framing it in my head, and you can probably tell me if I'm framing this incorrectly, that if the perception of neuroscience research today, I'm not an insider, I'm not a trained neuroscientist, but as somebody who consumes a lot of literature and a lot of the popular books that come out on the subject, which is, I don't watch the pop videos. I literally find the actual lab symposium videos and I sit and watch them because I think that's where the debate actually happens. The way I'm sort of framing it in my head is that neuroscience today is to a certain extent understanding where we came from. Why are we the way we are? Why is our neural computation the way that it is in? Why is that the evolutionary bedrock or the evolutionary framework that it is actually computing on? Now, if you look at the way the Neuralink or something like that comes in, where it becomes a completely extrapolated level of intelligence, where you take the sum total of progress in computers and electronics and all of that. And it's like the Matrix, right? I've just learned Kung Fu. That's the line that he says. And, what is your view, since we're touching upon the ethics and we're at the ethics segment of it, what is your general perspective in terms of intelligence augmentation in actual mammals as opposed to mere computer simulations giving you answers at your whims and fancies?
Yeah, this is one of those things that is at the edge of ethics that I really worry about. I'm close to 50 years old now, so I'm a little bit old fashioned. It makes me very uncomfortable. I think already, with all these large language models, we are losing out our edge on creativity, on imagination, because nobody is going to push themselves when they have a handy AI tool with them, which they can use. This is what I tell my students, for example, when we wrote papers, we would sit there with writer's block trying to get that one sentence, one perfect sentence, one perfect way of saying things, right? And then we would go through draft after draft after draft, and it would be a painful process. But what it does to you is that it makes you learn how to write, how to be precise, how to be specific, uh, and at the same time present something in simple language, which is all about scientific communication, right? And now what happens is that you have chat gpt, right? You throw in a few leading questions to it and you get a first draft. In worst of cases, there have been things on Twitter where people have shown that there are papers written by ChatGPT without even the authors looking at the first sentence, which says, here is something that you wanted. Even that has not been deleted, right? So it's going to make us dumb. That's one thing, right? And then you have this augmented intelligence, which means that first of all, what is intelligence? Intelligence is a very abstract concept. It's not something. There are different kinds of intelligences. There's not just one kind of intelligence. So when you say intelligence augmentation, what are you augmenting? Correct? And what is that going to be as effective as something that you do naturally, right? It comes because people believe that they can't augment intelligence. This is a very ingrained idea that I find in a lot of people, that they think intelligence is something that a person is born with. Wrong, right? There may be a few cases of gifted children, people with extraordinary ability, but we're talking about outliers here. for the most part of the population, intelligence is something that you develop by learning. Yeah. And if you're going to augment it the shortcut way, you're not putting in the hard work to get there, right? And not only that, um, it's going to make you one dimensional, right? So for example, if I want to become a piano maestro, okay, that's one kind of musically talented, musically intelligent. I'm going to put in the hours to go and train with a person who's a piano teacher, and maybe I will have social uh, interactions there. Maybe I'll make friends there, correct? But if you just plonk a chip on my brain, I'm just going to be playing a piano like a robot.
Maybe that's actually really interesting, because I completely agree with the social dynamic side of the argument. There's no denying that. But the way I am also sort of seeing it is that there is no guarantee that there's no set timeline of when the synaptic sort of updates will be from the brain to the tips of your fingers to actually play those notes on the piano. We don't know how long that is going to take. Okay.
Mm-hmm.
But what I'm sort of getting at is that probably you would be able to hear music a lot better, right? Or
Absolutely.
probably, you would feel like what there's a really interesting analogy that somebody gave, which is that, who lived a better life, the king of France in the 16th century or you
Hmm.
today, right?
Hmm.
The
Hmm.
technically correct answer is you today, right?
Correct.
It's because we have unlimited access to compute. We have, well, it's limited access, but in our pocket it feels it's unlimited, right? Yeah, I have to be very specific on these issues. So, you have a lot of compute. You have the ability to find resources, fetch resources. The cost of fetching it has gone down. Your ability to share these ideas has gone down. Your ability to promote and sort of come up in a quasi-egalitarian way has gone up, right? Versus being the king of France, who would probably die in his 30s or mid 40s, right? So
Mm-hmm.
the reason why I'm making this comparison is that I am not very, the way I sort of compare it to the piano player analogy that you gave is that probably within the next 50 years or 70 years, you will be able to hear music the way probably Beethoven heard it in his mind or
Mm-hmm.
inside
Mm-hmm.
his own head. The quality that
Right.
you have would be at that level, but it's still going to take you practice to sit and play the piano. So
Correct.
probably the closest scientific analogy I can get is probably Ramanujan, right? You will
Mm.
probably not need proofs. The
Mm.
equations simply manifest themselves, right? But you're still going to
Mm-hmm.
need to sit down and know what's the right question to ask. So I think augmentation is a great space because, for example, I don't know how to dance. I have bad rhythmic timing,
Mm.
but I am a trained musician, so I love music, right?
Mm-hmm.
So probably I'll be able to. I don't know how augmentation is going to work in that context, but
Mm.
that's the difference between augmentation and application. And it's. I just wanted to share that sort of a interesting distinction and see
Maybe
what you
you'll get
thought.
an exoskeleton that will allow, all you need to do is connect your
Absolutely right.
chip to your exoskeleton.
Absolutely. I think that's really interesting because, see, I think what's happened is there's this lovely anecdote I like to share with people. So there was a science fiction conference in China almost 15-20 years ago, and Neil Gaiman, the celebrated science fiction and fantasy writer, attended that conference. He went there and he was absolutely blown away by the progress that China made. And he asked, how is it that you guys opened up your economy only relatively in the 70s with your own version of capitalism and all of that, or socialism or whatever it is, but how is it that you achieved this level of innovation and sort of a scientific culture and focus? And the apparently the CCP person that who was accompanying him in that conversation basically said, we attacked it at the root of the problem. And he asked, what is that? He said, we made state sanctioned science fiction pictures and science fiction stories, so that the kids when they grew up wanted to be doctors and engineers as opposed to anything else. So the reason I'm saying that is because it's really interesting. You spoke about an exoskeleton, and how many times there's been a kid at school who says that, hey, I want to make an exoskeleton, but there's no reference to native exoskeleton stories that they can think of sort of doing, so they don't even have a rational place to start. So my question to you is this: what is the power of imagination when it comes to doing research or being curious in India?
Well, that's everything, right? I would say imagination is the bedrock on which research stands, because you're trying to do something or find something with whatever it is that you have, and trying to discover new knowledge, right? And to discover new knowledge, you have to be clever in framing questions, because if it were easy, somebody would have solved it already. And with every passing year, the bar just keeps getting higher and higher, right? And like I said, especially being in India, you have to think of interesting questions that the world will sit up and take notice, right? But at the same time work in Indian conditions, and with some sort of resource limitation, right? And so this requires a lot of imagination in your head as to where it is that I should invest my energy, where the questions are significant and it's feasible given my situation, my circumstances here. It's almost like, I would like to frequently draw analogy with the movie industry, for example. There are big banner movies, like Avatar, for example, and then there are low budget movies, and people watch both, right? And there are good stories to be told with both styles of movie making.
And bad stories.
um, and bad stories, yes. Hopefully we don't want to do that.
Absolutely.
So you want to be imaginative in creating a niche for yourself where you can ask the most interesting questions, and that requires tremendous imagination and creativity.
But there's like I agree, and I think I think you'll probably succeed at that for a generation, is my view. And the reason
Hmm.
I'm making that point very sort of carefully is because all it takes is one sort of a trend to really change the way people do something that's been honored and time bound and time honored traditions, right? The concrete example that I like to give is that there's a very famous block. So I actually read Freakonomics when I was in school. Okay, so Freakonomics, interestingly, was my entry to economics. I never took economics in any capacity whatsoever. But when there's this guy who's doing absolutely madcap sort of analysis of why drug dealers live with their mothers, and how it is basically structured like McDonald's, or it's even far more capital efficient than McDonald's, and you're like a 13-year-old kid, 14, you're like a 16-year-old kid, really like, this is amazing. And there was actually a professor who actually came out and said that Levitt has basically destroyed an entire generation of economists because they didn't want to come and do the sort of really critical micro sort of work. They wanted to find interesting correlational studies that had no basis in anything, like why do scissor producers basically have coal mines or something like that, or some
Yeah.
like.
This is very common in all fields, actually. Yeah, yeah, yeah. There's something that catches the imagination. So, for example, when fMRI came out, functional magnetic resonance
Yeah.
imaging, you had to stick a human into an MRI machine, make them do interesting stuff, image their brain, and then have a color map for which parts of the brain got activated when they spelled the word reward or whatever, right? They did tic-tac-toe, what part of the brain gets activated, and more, more interesting and titillating stuff as well. But there became a point where these papers were really becoming annoying, because the controls were not there. And as you said, they're not going and asking the more interesting questions, like, okay, given that these parts of the brain are activated, what are you going to do about it? How are you going to understand what really is going on in these parts of the brain, right? And we have another moment like that in neuroscience, where now we have imaging activity, calcium imaging. And people just go in and image the brain, while animal models do different things. A rat is running on a ball, you have a little microscope mounted on its head, and you're seeing, okay, this part of the brain is active, that part of the brain is active. um, and we have another tool in the box right now, which is optogenetics, which means that you can shine light on a particular part of the brain and make the neurons go on or off. And again, this comes with a lot of caveats, because if you turn on a neuron where it is normally silent and the animal does something, it doesn't mean that that's how the brain actually does the task. For example, let me say you have a mouse on a ball, it's not moving. You have a little thing on its head and you shine light and turn the neuron on, and the mouse starts moving on the ball. You can't conclude that normally when the mouse is moving, it is that part of the brain that gets activated, because redundancy, like I said before, brain has many ways of doing things, and there are connected loops and pathways. And so when you activate one part, you may simply be upsetting the excitation inhibition balance in the brain. You as an experimenter are doing something. It's like similar to Heisenberg's uncertainty principle. So this is sort of, I would say, retarded the field, because everybody is now in this business of imaging, and then using optogenetics to activate or suppress, and say that, okay, this circuit is doing this. And we are now just stamp collectors. We put the catalog of, okay, this part does this part does this, and so on. And it's not really led us to an integrative understanding of how the brain works. And I hope, it's like, what do you call it? It's like a reward thing, that you keep on pressing the button, that you're getting rewarded. It's like playing a video game and you're constantly hitting the targets. So you're always only engaged with getting the reward of publishing a paper, or saying that, okay, this is how this works, but you're not moving on to the next step.
That's actually really interesting that you brought up the imaging trend up in the conversation, because I don't know how much of a meme it was in neuroscience Twitter, but I distinctly remember seeing like one conversation between like a couple of postdocs on Twitter, and they were basically criticizing a couple of things that was getting published. I don't recall when this was, and one guy was like, I saw this paper and it was about XYZ, and they just imaged XY ABC, right? And the other guy below basically went like, is like, all these conferences have basically become just this one sentence. Hey bro, let me show you a cool imaging study. That's how, like, that's what it's become. He said.
Absolutely.
Yeah.
No, I mean, every field needs these kinds of moments, right? But it takes a fad, once it sets in, it takes a decade. Uh, there was a time when LTP, long term potentiation, was all the rage. Meaning, so long term potentiation is when the synaptic connection between two neurons becomes very strong, because that pathway is used a lot, right? When you stimulate that pathway, then it becomes strong. And this was touted to be the mechanism for memory. And so, people were mad about LTP, and everywhere you turned, it was about
So is
LTP and
this from where
neuroscience.
the the meme of neurons
Fire
fired
together.
together? Is
Yeah, yeah,
this
yeah,
where it
yeah,
came
yeah.
from?
But it was pre, I mean, it was before that. So to be sure, the two are separate phenomena. So this was Hebb's postulate. And Hebb said that, if two neurons fire together, then their connection between them becomes stronger. And LTP, on the other hand, is tetanic stimulation, which is very high frequency stimulation. It's not like a physiological fire together, wire together kind of plasticity. So the two are distinct, but it still is based on the fact that the two neurons are correlated in their activity.
Right. So I want to come to section three of our conversation, which is India
Mhm.
and the science of the future. I'm very careful in choosing those words, because my first question to you is, on in this particular segment, is you know what will the Indian scientist of the future look like in terms of what they do, where they work, and how they are enabled to do the kind of work that they do. What's the optimistic vision that you have for the future of the Indian scientist?
Yeah, so first of all, I think it will be much more geographically diverse. It's become like that in the last say 6 7 years. Earlier, it used to be few pockets in the country, especially Bangalore is a big hub, and the national capital area is another big hub. Bombay, Pune area is another hub. But now with the opening up of many institutes across the country, it's become much more widely located. So there are good institutes to be found in tier 2 tier 3 institute cities as well. The second thing is, I think we will have much more participation from the universities, both public and private. So, this is a good, healthy trend that I'm seeing, that there are many more private universities as well, where research is given a lot of priority in both the hiring of faculty as well as providing them with resources. So we will see much more involvement of the universities, and that will mean again, to highlight the positives, I think much more involvement of undergraduates, uh, which until now is not to be spoken of in great detail at all. So once you have a decent research infrastructure at universities, I think we can expect many more undergraduates to participate in research, and therefore have more youthful as well as more diverse entry population into science.
I think that's really interesting, ma'am. I'm only saying this because uh, we always look at the university laboratory ecosystem, the days of DARPA, as the origins of a lot of technological boom that we are enjoying today, right? But I think very rarely is it mentioned that it would not be possible had it not been for AT&T Bell Labs. I mean, Bell Labs specifically, right? Because at one point, or the RAND Corporation or whatever they do today, I'm not condoning anything, but back in the day when a Claude Shannon used to work at Bell Labs, right? Shockley was at Bell Labs, right? So, what is your view on the role of, because India has abysmal private sector participation in R&D. I don't know how many people I speak to generally. I tell them, or they know already, that India has a GERD of 0.7%. You know, and it's low, and the best performing countries have 2%, 3%. China's really pushing it up, right? Uh, the first question I sort of have is, what should the private sector do, especially in biotech and life sciences, to at least up the ante and drive it from from point 0.7 to at least 1%, given the following factors. A, you had a huge win with the vaccine initiative. Okay. B, you have a large swarm of young people with a demographic dividend specializing in triple biotech, and CS. Okay. So there's a lot of translational skills that actually exist if they are going to do life sciences research, right? They can come in and it becomes much easier because they bring that engineering mindset. And number three, what is the support of a role of mentors and senior scientists such as yourself, and the government that is actually required to actually
Hmm.
ensure that we create a new crop of ultra competent and incredible scientists doing work in, say, life extension or something like that?
Absolutely. So I'll try and remember all of the questions that you asked one by one. So first, for the percent GDP that's being spent on R&D, yes, it is abysmal. Um, you know, you can't think of, there's all this talk about 2047 developed economy and all that. R&D is a key foundation on which the economy is built. You cannot not spend on R&D and hope to be a developed economy. So that's something that needs to be fixed. But even before we go into increasing the expenditure on R&D, is something to be said about um, administering the money that you currently have appropriately, right? So this is something that needs to be fixed that needs to have been fixed 10 years ago. It's still not fixed. Um, what we're losing out is human power, brain power. Brain drain is happening on a massive scale. There is no dearth of very good undergraduate institutions in the country. We have the ICERs and the IITs, IISc, for example. Brilliant students graduate out of these institutions, but most of them go abroad, you know. Um, and one of the big reasons they go abroad is that there is not even a guarantee for their living expenses here in India, right? Their fellowships don't come on time. Um, and if the fellowships don't come on time, then how do you expect a person to live and support themselves? And that's one question. But the next point is also that that shows you don't respect them, right? And who would want to remain as a PhD scholar in a place where their contribution is not being valued, right? So this is one thing that needs to be fixed pronto. Um, Many people have voiced their opinions on this. Many top scientists have said that this needs to be fixed. I don't know where the hiccup is, or what the problem is, and why we can't just have dispersal of funds on time. Same thing happens for grant money dispersal, where you submit a letter of intent. It takes 1 year for the grant full proposal invitation to come. You submit a full grant proposal. It takes several months again to hear from the grant agency as to what happened to the grant. And even if you get approval, there's no clarity on when the money will come for the first year. Even if the money comes on the first year, funds are delayed in the second and the third year. Many times, the full amount is not given. You ask for X amount of money, it's arbitrarily reduced. Maybe it's not the problem of the granting agency. Maybe it's that the fire, they just don't have the money to give to people. So these are operational, purely operational and administrative things. If these are ironed out, it will actually make doing science in this country much easier. The government talks about ease of doing business, right? Unfortunately, nobody talks about ease of doing research, right? Because we are not thought of as a field that generates revenue. But what people forget is that, it's not an short term revenue generating field. Research is something that you invest in for the future. Um, all of the vaccines that you talked about, the COVID vaccines and things, came out because of research that was done on mRNA, on mRNA vaccines, many years before even somebody thought of making it into a product, right? Many biological discoveries that are biomedically important, that have proved to be commercially very important, came out not because somebody said that, I want to make this product or I want to cure this disease. People were just doing curiosity based research and they found something. Even the original discoverers didn't imagine that their discovery would have something to do with human health. Case in point, CRISPR technology, which is used for genome editing. When the original research was done, they were looking at bacterial immunity to viral infections. Who would have thought that it would have an implication for genome editing later, right? So it's very short sighted, and it will reflect our hubris if we think that we can only do research that is economically going to reward us immediately, right? This is something that people have to have much more of an open mind on, uh, and try to open up the purse strings for more blue sky research, because that's what will lead us into the future. So that's one thing. And the third point about the private participation. So private participation in this country is driven by this philosophy that, what's in it for me immediately, what can I make as a product out of this. I believe that's changing. We have more corporate social responsibility funds, CSR funds, that are now being utilized for research purposes. More companies should come forward to do that. That should be a mechanism for all research centers to be able to draw on the CSR funds. and also make it easier for us to access these pots of money. That'll be good. Yeah.
No, I think that's really interesting that you brought up the reason why I brought up the private sector point was because there is always a very popular notion that Google's culture is the way it is because it is an extension of the founders' graduate school experience at Stanford, right? So America has this really interesting relationship with funding science because it was first state driven. It's still state driven. DEA is still the largest donor of funds for actually running research in the US, right? The Atomic Energy Department. Uh, the interesting thing that I see is that whether it's an Nvidia or it is a Google or it is an Apple or a Microsoft or any of these companies, the corporation has certain goals. The goals can be long term goals, and the goals can be in synergy with what the researchers want to achieve. So they meet halfway. They at least have a conversation to meet halfway. And I think our corporations are not doing that. Our companies are not doing that because I think we're still at that early stage of the early Google years, so to speak. The second point I want to add is that it's very interesting, Dr. Vatsala, that um, a large swath of science is fundamentally driven by the question is why is it that we spoke about talent, right? The talent and the brain drain that you actually referred, if you look at the top, there is a incredible conference called NeuroIPS, right? NIPS, it's called NIPS. Uh, the top 5.5% of highly cited papers in that were actually written by Indians. Interestingly, hardly any of them are actually in India. So the question is, you are creating an ecosystem where you have exceptional demographics, exceptional talent, really doing well, working hard. The question is, where is the commitment from the
Hmm.
private sector and the government to at least have a conversation and meet halfway?
Right, right. So, for example, the bright sparks are that there are small initiatives and a little bit relatively larger initiatives like Chris Gopalakrishnan's Pratiksha Trust is funding a lot of neuroscience research. They have this Center for Brain Research. Rohini Nilekani's Center for Brain and Mind, and here in Chennai, IIT, there is a Center for Brain Research, mostly funded by Infosys founders. I hope other companies also take note, and they do fund fundamental research, not just in neuroscience, but all of the frontier sciences. But I think it will take probably like the Google story. If there is a unicorn that comes out of a campus, and then people will it will catch on, right? That they will see that you know PhDs are valuable because they made something that could be profitable. So that has to happen. But there are, I mean, given the number of startups that are coming up now, it's only a matter of time. Maybe there'll be a unicorn that
Absolutely.
comes out of campus and is appreciated.
Absolutely. And I just want to sort of come back to the role of mentorship. I think it's very important that young scientists know that there are incredible mentors in India to guide them on their journey. So, can you talk a little bit about a few mentors from your career and the role that they have
Absolutely.
had on you?
Yeah, definitely. I would say my strongest mentor has been my PhD advisor, Professor Eve Marder. Uh, there's a lot of things I learned from her, um, including how to mentor people. So she has this philosophy that different people require different styles of mentorship. You can't be the same person to everybody because their mentorship needs are different, and that's something that I practice from day one. Um, there are some people who may require your reassurance and hand holding a little bit, whereas some other people would take that as an insult. So you have to be a little bit sensitive to how you mentor people based on their mentorship needs, and it's it's not easy to figure out, right? It takes a certain amount of perception, knowing their personality and their background and things like that. And I don't claim to have got it correct all the time. You know, everybody makes mistakes. um, the other thing that I learned from her was to always appreciate the complexity of the unknown, to not have hubris over what you know, because the unknown is always is a little bit more complex than what you think it is. Um, the third most important thing that I learned from her is not to do what she calls consensus science, which is people, there's a science problem that everybody agrees is important. You don't necessarily have to work on something that a lot of people think is an important problem just because there are a lot of people think it's an important problem, right? You can define your own problem that you think is important. You don't need a crowd to tell you that what you're working on is important. So this is something that you know uh, I keep in my mind, in terms of trying to find the question that interests me most, regardless of what other people might think about it. So that's in a nutshell. But yeah, I mean, I've had great teachers in school. I had a postdoc mentor who gave me independence to do whatever I wanted. That was a big confidence building exercise, so that was good. But to this day, Eve holds a special place.
And I think that's a wonderful place to put a bookend on our conversation. So, Dr. Vatsa, thank you so much for your time and patience. I deeply appreciate it. Thank you so much for sharing your insights, your work, your ideas. And, I'm sure that there are a lot of people who will be listening to this podcast who share your sense of optimism, your frustrations, and probably, down the line, we might even figure out a way to collect all these challenges and ensure that we have an ease of doing research, sort of an approach that will help us become the, there's a very famous phrase that, we've been the back office of the world, right? I think it's time that, we understand what the problems are and we move from the back office of the world to the boardrooms and the labs of the world. So thank you so much for your time and patience, and I deeply appreciate it.
Thank you, Vignesh. I really enjoyed this conversation.